Cerebral palsy is a disorder of posture, movement, and muscle tone. It is an upper motor neuron injury and is commonly caused by an injury to the developing brain of a fetus. Symptoms include, but are not limited to, hypertonicity, ataxia, spasticity, and athetosis. Common complications associated with cerebral palsy are contractures, osteoarthritis, osteopenia, and a variety of mental health conditions. For our project, we focused on cerebral palsy and how the physiology of muscle stretching is different in this population from the physiology of muscle stretching in a healthy population.
We will briefly go over some objectives that we wish you take away from the video after watching. Number 1, students will identify the characteristics of cerebral palsy. Number 2, students will understand the general mechanism of the muscle stretch reflex and how it differs in patients with cerebral palsy. Number 3, students will understand the physiological limitations of a muscle in a person with cerebral palsy. And last, number 4, students will know the proposed effects of stretching on neurological and mechanical aspects of hypertonicity.
Although we've gone over these definitions in a few other classes, here are some key terms that may be helpful in understanding the disease a bit more. Hypertonicity is an increase in muscle tone and an increased resistance to passive stretch. Spasticity is an increase in reflexes and resistance to passive stretch that increases as the velocity of the stretch increases. A contracture is the shortening of a tissue that causes a deformity. An athetosis is abnormal muscle contractions that cause writhing movements. Because the severity of the disease differs from person to person, cerebral palsy is often graded using the Gross Motor Function Classification System, or GMFCS, which has levels 1 through 5. Level 1 can run, jump, and climb stairs, but they have decreased coordination, balance, and speed. Level 2 can walk and climb stairs, but may have difficulty in open environments and may need an assistive device for long distances. Level 3 can walk using an assistive device indoors, but may require a wheeled device such as a wheelchair for longer distances. Our research focused on levels 1 through 3, as these are the levels that can ambulate independently with or without an assistive device, and that was important for some of the interventions we looked at. It's also important to note that it is unlikely that the GMFCS level will improve past the age of 5, since our populations were over this age.
Our skeletal muscles are made up of extrafusal and intrafusal fibers. The extrafusal fibers are the ones we typically think of that cause a muscle contraction and are innervated by alpha motor neurons. However, the intrafusal fibers also play an important role in muscular function. The intrafusal fibers, also known as muscle spindles, detect the level of stretch placed on a muscle to help the central nervous system understand its location in space and can react to stretches to prevent injury. During a stretch reflex, type 1A and type 2 afferent fibers send a signal to the spinal cord to indicate that the muscle has been stretched. The rate of firing is dependent on the magnitude and speed of stretch. Type 1A fibers are sensitive to changes in length and velocity, while type 2 fibers are only sensitive to changes in length. These afferent pathways synapse in the spinal cord and cause activation of gamma and alpha motor neurons. The gamma motor neurons will adjust the length of the intrafusal fibers to accommodate the change in length of the whole muscle. and the alpha motor neurons cause muscle contraction, or muscle shortening. In children with cerebral palsy, there is a loss of descending control that can modulate this reflex pathway. Without connection to the reticulospinal tract, the type 1A and type 2 afferent fibers become more sensitive to stretch and fire rapidly and unnecessarily. This causes an increased recruitment in alpha motor neurons that will contract the muscle in an inappropriate amount. Another mechanism for increased muscle tone in patients with cerebral palsy is the changes in muscle characteristics. Typical muscles contain satellite cells that are stem cells that will proliferate in response to muscle damage and then initiate muscle regeneration and generally promote muscle growth. With a lack of satellite cells, the muscle does not grow adequately with development and does not remodel the same way as a normally developing muscle would. More specifically, satellite cells are required for the remodeling of the extracellular matrix, which is responsible for maintaining a passive and elastic response of the muscle and allows for a lateral force transmission between neighboring fibers. The extracellular matrix is composed of several structures, including collagen. Collagen is responsible for the tensile strength of the structure. Its stiff nature makes it resistant to deformation. However, in children with cerebral palsy, there is a lack of these satellite cells that can organize collagen content. The deficient quantity of satellite cells can inhibit normal muscle growth, creating a shorter muscle length in these children. Additionally, research has shown that patients with cerebral palsy have longer sarcomeres, but a shorter quantity of them in series. This makes the muscle itself shorter. Therefore, there's a buildup of collagen in these muscles that make them physically stiffer. Similarly, the absence of satellite cells could be a mechanism for increased collagen levels. The high levels of collagen increase muscle stiffness. The combination of morphological stiffness and neural overactivation leads to hypertonic muscles that can be hard to control and make activities of daily living far more challenging.
Now that we've talked in more detail about what components make up the ECM, we can talk about how those components are regulated by their physiological factors. One of these factors is TGF-beta 1. Collagen turnover is controlled by TGF-beta 1. This is a cytokine which plays a role in activating the gene expression for synthesis of ECM components. It has been shown that TGF-beta 1 mRNA is upregulated in people with cerebral palsy. Another physiological factor is MMP1. MMP1 is a protein-coding gene that is needed to begin the cycle of collagen turnover. It works by cleaving the collagen into smaller gelatins so that they can be degraded more easily. There were decreased levels of MMP1 found in the cerebral palsy population compared to the healthy population. However, the researchers noted that even in the healthy population, there was a large range of MMP1 levels, meaning that these levels differ from person to person, even in a healthy population, making them a little bit more difficult to work with in research. This research study also found that patients with more involved CP, such as those with quadriplegia, had higher mRNA levels of these genes than those with diplegia. However, there is more research that needs to be done to substantiate this relationship that these researchers found.
We can use this picture to reiterate the changes along the muscle stretch reflex pathway that result in increased muscle tone. When a stretch first occurs in healthy individuals, type 1A and type 2 fibers detect the change in length and send a signal to the spinal cord. The connection to the central nervous system is still intact, so the reticulospinal tract can modulate this pathway. An appropriate amount of alpha motor neurons respond to the muscle stretch, cause a contraction, and restore the muscle's original length. In patients with cerebral palsy, the connection to the reticulospinal tract is lost, so there is no modulation of the muscle stretch reflex. The sensitivity of the type 1A and type 2 fibers increase, so they fire more often. As a result, many more alpha motor neurons are recruited and the muscle contracts to a greater degree. This flawed reflex pathway is activated very often in patients with cerebral palsy, which contributes to their muscle hypertonicity.
Here we have Sam. Sam was born with cerebral palsy and has been going to physical therapy since he was young. Today we are going to demonstrate some of the stretching interventions that Sam has been participating in when he sees his PT. As soon as Sam enters the clinic, he is greeted by his physical therapist, and they soon begin the interventions that they typically do on an average day. Today, we're going to go through some of these stretching techniques that the physical therapist uses. We are also going to include studies and articles that show how these stretching techniques are reviewed in the literature and the outcomes seen in typical studies. A common treatment used in children with cerebral palsy is stretching. There are many different ways to stretch. However, the most common method in patients with cerebral palsy is static stretching. It can be used on any muscle that is experiencing spasticity or contractures, but many studies focus on the lower extremity, with the large muscle groups in the thigh and lower leg being a major focus. In this first scene, we are demonstrating the technique that Wart et al., who reviewed 7 studies, including a study by McPherson et al., discussed that looking at prescribing 5 times 60 seconds of hamstring stretching at maximum tolerance without pain. However, Wart et al. Noted that the evidence behind stretching can neither support nor refute its use in treatment. In the second scene, we are demonstrating functional stretching that Palsy et al. Study that discussed utilizing functional stretching was an effective treatment for spastic diplegic children when applied to hip flexors, hip abductors, hamstrings, and calf muscles. In order to achieve this functional stretch, the participants maintained a standing walking position with one leg forward and one leg in what they called a stride stand position, which involves standing with the feet wider than shoulder width apart. In both positions, a therapist is providing support to the legs so the patient maintains the stretch and does not alter the position. These stretches prescribed at 3 to 5 times 30 seconds with about a 30-second rest between reps was found to improve mobility as evidenced by an increase in popliteal angle and improvement in gait after treatment. It is believed by integrating sensory stimulation by stretching during functional activities, it allowed for better learning processes and function. While maintaining flexibility is a critical component of CP, and stretching is a common method to do this, the functional stretching utilized by Palsy et al. In the third scene was definitely better than the passive stretching demonstrated in other studies. In the third scene, we are demonstrating what Wart et al. As well as Wu et al. Studied when looking at the effects of passive range of motion and active range of motion on spasticity. Subjects in the Wu et al. Study had participants receive 20 minutes of passive stretching, followed by 30 minutes of active movement stretching, which was considered to be the same as active range of motion, and ended with 10 more minutes of passive stretching. This was performed in plantar flexion and dorsiflexion, and it was found that there was increased joint mechanical properties, motor control performance, and functional capacity in balance and mobility. Just like Palsy et al., it is believed that the improvement in motor learning was achieved through the active process. While Wart et al. Concluded that stretching was ineffective, they only examined passive interventions. It appears that more functional and active participation by the patient will result in better outcomes. In terms of treatment, this means that it may be a good idea to incorporate stretching, as long as the treatment session involves active participation by the patient. These stretches shown here today are a few that studies have looked at for interventions in the stretching components. PT will often take their patients through these types of stretches on a daily routine and often encourage this to be done at home. Physical therapists may also give them exercise for strength training and postural stability. Today, we chose to just look at some stretching interventions, and now we'll take a look at how a healthy population responds to stretching versus a patient with cerebral palsy.
In a relatively healthy person, there is a lot of physiological components to the mechanism of a stretch. The main goal of a stretch is to improve flexibility. Within the ECM, we have elastin and collagen. Elastin, which provides the elasticity of a muscle, and collagen that provides the tensile strength. When a person stretches, these components are put under tension, which allows them to deform and elongate. Satellite cells are spread throughout the muscle and maintain its health. These satellite cells regulate collagen and differentiate to help the muscles grow. With properly functioning satellite cells, the muscle maintains proper collagen content, which limits the increased stiffness and decreases the likelihood of a contracture. Intrafusal fibers like Golgi tendon organs and muscle spindle fibers will fire if the muscle is stretched and allows the muscle to use proper force for contraction and protects from a tear. A stretch should be performed slowly to prevent intrafusal fibers from limiting the range of motion by signaling muscle contraction. Repetitive stretching can increase the firing threshold for these intrafusal fibers. The primary mechanism for the overall lengthening of a muscle is creep. The constant load placed on the muscle will create deformation. This consistent deformation allows for total muscle length gains through the previously mentioned muscle components. Within the contractile tissue, mucopolysaccharides are lubricating molecules that allow the fibers to easily glide across each other during movement. There is also another proposed mechanism that may be responsible for total muscle length gains. This mechanism is called sarcomerogenesis. This is when new sarcomeres are created in series due to passive stretching. This can elongate the muscle fibers to optimal length and create a more favorable position for muscle contraction. Most of the studies showing sarcomerogenesis have been done on animal subjects, but there is evidence that this could be a biomechanism in humans as well. In many studies, including Zollner et al., sarcomere length decreases over time, while the number of sarcomeres in series increases. This could be an important mechanism to determine the effects of a stretch within impaired muscles. But what happens in the muscle of a person with cerebral palsy? Like we said earlier, people with CP have hypertonicity, which is a resting resistance of muscles to change in length. This hypertonicity can lead to muscle contractures that limit range of motion and create muscle stiffness. The extracellular matrix and collagen play a distinct role in creating this stiffness. Satellite cells regulate the collagen within the ECM. Studies have found that people with CP have a significantly lower satellite cell content compared to typically developing people. Studies have also shown that children with cerebral palsy have higher collagen content within the extracellular matrix. This could be related to the decrease of satellite cells. This collagen content creates more resistance to deformation compared to typically developing children. It has also been shown that people with CP have significant increase of sarcomere length. This increased collagen content within the ECM, combined with the sarcomere length, create the significant passive stiffness that we can notice working with this population. It is hypothesized that passive stretching is an effective intervention to reduce the risk of contractures, hypertonicity, and spasticity in people with cerebral palsy. However, there is a lack of research surrounding this hypothesis. It is thought that the muscle will have both morphological and neuromotor responses to the stretch. The morphological responses include an elongated extracellular matrix due to the concept of creep. Collagen fibers are thought to align properly along the line of pull due to the mechanical stress. Overall, this should reduce the risk of contractures with these two concepts combined. The neuromotor response would stem from the intrafusal fibers. The intrafusal fibers will be activated by the stretch, inhibit the excitable alpha motor neurons, and therefore increase the extensibility of the tissues. The neuromotor response should help reduce hypertonicity and spasticity in the patient. In order to take a deeper look into the physiological response of stretching for people with cerebral palsy, we have analyzed some research. Wu et al. Did a study using passive stretching combined with active movement training with a portable robot. The participant was strapped into a robotic boot that allowed them to receive passive stretching and do active range of motion activities that were measured by the robot. There were many notable results from this study. These include a significant increase of dorsiflexion passive range of motion and active range of motion, decrease of joint stiffness, and increase of dorsiflexion torque production. The increase of passive range of motion and decrease of joint stiffness may have influence on the increased torque production and ankle mechanics. Passive stretching in combination with active range of motion training can improve biomechanics, motor control, and overall function. The article was the main source behind our combined active range of motion and passive range of motion stretch. McNee et al. Did a study with children with cerebral palsy to determine the impact of serial casting on gait. The participants' ankles were casted at maximum passive dorsiflexion. The cast was replaced each week until there was no range of motion increase or the participant had met the dorsiflexion goal. The thought is that immobilization could induce sarcomerogenesis, or the growth of new sarcomeres in series, which would lengthen the muscle belly. The study found a significant increase of dorsiflexion range of motion. However, prolonged immobilization could lead to muscle atrophy, which would be detrimental to the patients, especially in this population. Overall, the use of casting has its disadvantages, but we are able to still see the effects of stretching on range of motion. Another important component of cerebral palsy is the neural effects. Thies et al. Did a study to determine the effects of stretching with cerebral palsy. The authors explained that hypertonicity has two parts, neural hypertonicity and mechanical hypertonicity. We know people with cerebral palsy are hyperreflexive due to decreased CNS modulation. We can use stretching to both decrease the reflexive activity and increase the threshold of the stretch reflex. The combination of these effects can reduce the excitability of motor neurons, reduce neural hypertonicity and spasticity, and therefore increase range of motion and muscle length. On the other hand, acute passive stretching can impact mechanical hypertonicity as well. Stretching can increase the length of the fascicle, increase the connective tissue length, and increase the number of sarcomeres in series through sarcomerogenesis. The result of Thies et al. Showed that there was a significant increase of dorsiflexion, tendon length, and muscle length with an acute passive stretch by PT and a self-stretch.
A study done by Smith et al. Looked at different parts of the muscle of children with cerebral palsy to determine what element was altered that allowed for contractures. The researchers looked at a biopsy of the hamstring in both children with CP and children without any neurological condition. From a biopsy of the hamstring, they found that titin was the same in both populations, and the individual fibers themselves were not altered in stiffness. Therefore, these elements were not indicated in the cause of the contractures. On the other hand, increased amounts of fibrillar collagen type 1 was found in both the semitendinosus and the gracilis of the CP subjects. It is important to note that the collagen in the subjects with CP was not found to be in a different arrangement than the collagen of the healthy population. Those with CP just had higher amounts of the collagen and a higher frequency of collagen deposits. Another interesting note is that the amount of another component of the ECM, called laminin, was also increased. Laminin is a structural protein that allows cells to attach to the basal lamina. It is sometimes referred to as the scaffolding of the tissue. This is an image from the Smith et al. Study in which they stained the ECM with an antibody to fibrillar collagen type 1. The pictures on the left are the control subjects, and the pictures on the right are the cerebral palsy subjects. Using this immunohistochemical technique, you can see the differences in the amount of ECM in those who have contractures secondary to cerebral palsy and the healthy population they were compared to.
This concludes our presentation for today. We just wanted to end on a few summary points that we wanted you to take away after seeing this presentation. Number 1, cerebral palsy is an upper motor neuron injury that influences posture, balance, and muscle tone. Number 2, according to research, functional stretching is more effective than static stretching in patients with cerebral palsy. Number 3, the increased sensitivity of type 1A and type 2 afferent fibers increased recruitment of alpha motor neurons in patients with cerebral palsy. Number 4, decreased satellite cell populations and increased collagen levels are limiting factors in muscle length. And lastly, number 5, stretching can reduce risk of contractures, hypertonicity, and spasticity by elongating the muscle and reducing the alpha motor neuron excitability. To make sure you got everything in this video that needed to be covered, we have made a Kahoot for everyone to engage in. So we put the code on the screen, and go ahead and put that code in and just complete the quiz and see how you do. Thanks for watching, everyone.